I think it was on conductive tissue. We learnt about SA node, AB node, huh? And bundle of feeds, also Purkinje fibers. But probably I didn't explain how cardiac impulse is generated. The competency is like this: generation of cardiac impulse and transmission of cardiac impulse through junctional tissues or conductive tissue to the heart muscles. That is the competency. I have explained what are these junctional tissues, what are these conductive tissues. Means SA node, AB node, bundle of fees, its branches. Now I came for this part, cardiac cycle. So that part is incomplete. Let me first complete that part. They have various electrical properties. There are mechanical properties, morphological properties, conductivity properties, and metabolic properties. The important electrical properties are of cardiac muscle. Important electrical properties are excitability, then automaticity. Contractility is the mechanical property. Conductivity. And more importantly, which is specific for cardiac muscle, it is automaticity. So automaticity, excitability, automaticity, and conductivity. Okay, these are also, there is another property, rhythmicity. Automaticity and rhythmicity together we will be explaining. That means autorhythmicity. So excitability, autorhythmicity, and conductivity. Excitability. What are excitability? Excitability is that electrical properties by which we mean the cells, if they are capable of responding to a stimulus, then it is called the cell is having excitability. That means cells should be capable of responding to a stimulus and thereby producing a response. So, firstly, they should be excited by stimulus and they should give a response. Okay? So, if I apply a stimulus, nerve cells are having excitability. They are excitable. Muscle cells, be it cardiac muscle, be it smooth muscle, be it skeletal muscles, they are having excitability. Means if we apply stimulus, they will be responding. Cardiac muscle, it will respond by producing action potential. Smooth muscle, they will be responding by producing action potential. And also, what are the other cell? Smooth muscle, cardiac cell, nerve cells, they will also produce action potential if they are stimulated. Okay, so now this action why this excitability? Because they are having a transmembrane potential, means resting membrane potential. If there is no resting membrane potential, the cell, it will not be possible to excite the cell. Okay, so cells having excitability, they respond to a stimulus by producing an action potential. Now this action potential in case of cardiac muscle are of two types. One is slow action potential, another is fast action potential. Now the cardiac muscles, heart, these are having three types of cells. One is, we call it working cardiac muscle. Means if you stimulate them, they will respond by contraction. There are another type of cells. They are called conductive tissue or junctional tissues of the heart. They don't contract. Instead, why they are in heart? They can produce cardiac impulse and they can transmit it to these signals. They can produce cardiac impulse and they can send this cardiac impulse through the special tissues to the cardiac muscles. And cardiac muscles, the working muscles, they need stimulus. And this stimulus will come from these cells who will be producing action potential, cardiac impulse. Okay? Two types. Another type of tissues are there. These are vascular tissue. What are these? Already you have learnt it. It is present within the heart, not outside the heart. What are these? What are these vascular system? Coronary circulations. So within the heart, what you will get? Cardiac muscles, coronary circulations. Again, cardiac muscles are two types. One is working muscle, another is special type of cells. Means these are junctional tissues, or another name is there. These are conductive tissue. Okay. Now let us see how the fast action potential is generated in working muscles. Again, I am telling you, what are working cardiac muscle? The cardiac muscle, when stimulated, which can contract, which can generate force. Okay, but this conductive tissue, they cannot do it. They are different from this. Now see, for their contraction, this working muscle, they need impulse. They need to be stimulated by the cardiac impulse. When they are stimulated, these cells are stimulated, what happens to them? Means they will be producing an action potential. How this action potential generated in the working cardiac muscle, you first learn it. Then it will be easy for you for learning how slow cardiac potential or cardiac impulse is generated in the SA node, junctional tissues, or in conductive tissue. We said two type of action potential is generated in the heart. One is slow, another is fast. Now we will be discussing fast, and this will be exhibited by which cells? Working cardiac muscles. Okay. Time. Millivolt. Resting membrane potential of working cardiac muscles. What is the resting membrane potential? Minus 80 millivolt. Okay. That means inside the membrane, there is more potassium ion in relation to outside, and in outside there is more sodium ion, more calcium ion. Okay. And net negative charge is inside more and net positive charge is outside more. And when you place two electrodes, one inside and one outside, and connect both the electrodes to a galvanometer, you will find a deflection. And how much? It will be showing some voltage difference. What is the voltage difference? Minus 80 millivolt under resting condition. So my ventricular muscles are in resting condition, having transmembrane potential of minus 80 millivolt. Now, cardiac impulse came to the ventricular muscle. Now what will happen? This is a muscle, working muscle. Outside is positively charged, inside negatively charged. Through circuitubular system, cardiac impulse came through junctional tissues to the cardiac muscles, working cardiac muscles. So there will also be a junction, cardiac muscle to junctional tissue end. So in this junction, when this impulse comes, this is cardiac impulse. Cardiac impulse means action potential, which is generated in SA node. It was produced in SA node. Through SA node, through junctional tissues, it is ending in ventricular muscles. Now, the ventricular muscle will respond to the stimulus. Means cardiac impulse coming from SA node. What will be there? Channels. There are channels. Sodium channels. Now the sodium channels will open. In presence of the stimulus, sodium channels will open. So outside sodium ion concentration is high, inside is less. So lot of sodium ion will be entering within the cell. Now what will happen? Sodium ion is entering with its positive charge. So when it is entering within the cell, the negativity will be neutralized gradually. So the difference, potential difference will be gradually become less negative. So there will be potential difference towards this or towards this. Whether it will be minus 85 or 90 or it will be minus 75 or 70. What? Which direction? Yes, less negative means in this direction, there will be potential change in this direction. So, I have given, it has been given stimulus here. Suppose this is the point where stimulus is given, arrived in the here. So, what immediately sodium ion started entering into the cell. So, there will be depolarization. So, the cell started giving depolarization slowly. When this depolarization came to this value, what happens? Immediately, some sodium channels which are voltage gated, they will open. What are these channels? Voltage gated channels. Now these voltage gated channels, opening of voltage, because of opening of the voltage gated channel, lot of sodium ion will enter into the cell. Lot of sodium ion will enter within the cell. As a result, there will be change in minus 75, minus 70, minus 60, minus 50, minus 40, minus 30, minus 20, minus 10, 0, plus 20, plus 10, plus 20, plus 30, change. So this will reach up to this. When it reaches up to this, because of sodium ion entry, sodium channel closes. This voltage gated sodium channel closes. Though there are still lot of sodium ion that can enter, but they cannot enter. Why? Because the channel is closing. Channel has become closed. Okay? So channel is closed. Why this change occurred? Because of rapid entry of sodium ion. Voltage gated sodium, opening of voltage gated sodium channel. Now what happens? What happens? When it reaches to this level, this change of depolarization is called, this change in voltage towards positivity is called depolarization. And this phase is called, this is a phase, this phase is called phase 0. And this phase, the resting phase, it is phase 4. The resting phase is phase 4. Okay? Now what will happen? We can see when it reaches here, immediately the voltage difference is coming down. It started coming to the reverse direction. Means towards the negativity, changes towards the negativity. Something, sodium ion is not entering, something coming out of the cell, means potassium ion is coming out. Since potassium ion from the cell, from the cell is coming outside, so it is coming out with positive charge towards outside. There is efflux of potassium ion. As a result, inside will start making it negative. Okay, so there will be a negative deflection. There will be negative deflection. Why it is? Because of entry of potassium ion within the cell. And this is called phase 1. Negative, slight negative deflection. Now what will happen? Calcium channel, lazy type, L type of calcium channel. There are two types of calcium channel present in cardiac muscle. One is T type, another transient type, another is L type. L type means lazy type. So now the lazy type, it's called lazy. Why? It opens slowly, it closes slowly. It takes time to open and takes time to close. So that's why it is called, this sort of channels are called lazy type, L type of calcium. Now there is calcium channel opening. Now what happens? Calcium ion is entering within the cell from outside with its positive charge, and potassium ion coming out with its positive charge. So entry of positive charge by calcium ion and exit of positive charge by potassium is same. Entry of positive ion, exit of positive is same. As a result, voltage change is not net, there is no net change, so we'll get a plateau. So we'll get a no change in voltage difference. It happens here at 0 level. So this will continue like this, and this phase is called phase 2. What is the cause of this phase 2? Why this plateau? Why this plateau? Mabashar, what is this cause of this plateau? Why this plateau? Why the voltage is not changing here in phase 3, phase 2? What is the cause of this voltage difference, this plateau?
The influx of calcium is equal to the efflux of potassium.
As a result,
No net change.
no net change in voltage. No net change. So in this way it will continue. And it continued up to this level. Thereafter, again the voltage is changing towards negativity. This phase is called phase 3. Phase 2, why it has started? Because here what happened? Closing of L-type calcium channel. But potassium ion is, efflux of potassium ion is continuing. As a result, there will be change towards negativity. So it is coming down. And gradually, what happened? It is slowing down and will coming back again to resting membrane potential. Again to resting membrane potential of minus 80 means phase 4. Again to phase 4. Now one thing happened. Lot of calcium or sodium ion has entered. Okay? They were outside. Now they are trapped within the cell. Lot of calcium ion has entered. Now they are trapped there. They trapped there. Lot of potassium ion came out. Now they are to be again reshuffled. They should go back to their original position. See what happened here. Calcium ion started entering. Why this calcium ion has entered within the cell? Because without calcium ion, the contraction is not possible. This calcium ion will cause contraction of the cardiac muscle with this calcium ion. So, lot of calcium ion has entered. Now it has stopped. So, by this time, when it has entered, the muscle started contracting, getting this calcium ion. So, here contraction is going to be finished, over. This action potential is over, contraction is also over. And here what will be happening? In phase 4, redistribution. How this redistribution will be made? By sodium potassium pump and by sodium calcium pump and by sodium calcium exchanger. So they will help in redistribution of the ions. যেটা ঢুকেছিল সেটা আবার বেরিয়ে যাবে, ওকে, যে যেখানে ছিল সেটাকে আবার সেন্ড ব্যাক। যেটা ক্যালসিয়াম আয়ন ঢুকেছিল from a sarcotubular system and sarcoplasmic reticulum, again they will go back to sarcoplasmic reticulum and to sarcotubular system. Okay, so what are the different phases we saw here? Phase 4, then phase 0. Phase 4 is nothing but resting membrane potential. Okay, and phase 0 means what happened there? Sodium initially, sodium entry slowly, then rapidly. When threshold limit is reached, then lot of voltage-gated sodium channel opened, lot of sodium ion entered, that make this upward stroke, upward change towards means depolarization. And from here, from this point to this point, this change is called repolarization. So this is depolarization and this is repolarization. So in repolarization we get phase 1, phase 2 and phase 3. Okay, so why repolarization? Initial part of repolarization, what is the cause? Exit of potassium ion. And what is the cause of phase 2? Entry of calcium ion and exit of potassium ion. And what is the cause of phase 3? Closing of calcium channel, and that remains always open, so calcium ion is going out. As a result, there is repolarization. Okay, and ultimately when it is coming to the resting level, then what will happen? The ions will be again go back to the original position, means there will be redistribution of ions. Okay, now this cardiac muscle is now ready to take another stimulation. Now it is ready to take. So if I want to make cardiac muscle give repeated stimuli, and we were expecting it will also be contract repeatedly, as well, what can happen? Cardiac muscle may very soon become fatigued, as you have seen in our last experiment. They will be fatigued, but we know cardiac muscle are not fatigued. Why? Because of their long refractory period. So during this refractory period, during this plateau period, if you apply a second stimulus to the cardiac muscle, it will not respond. So this is the absolute refractory period. If you apply a stimulus here, when this process is going on, if you apply a second stimulus, this will not produce any effect, because effect of first stimulus is going on, and the change is there in the muscle is now in refractory period. It will not respond to the second stimuli. But if you apply a second stimulus to this point, means this level, it is now, if the stimulation is strong enough, it can produce again from here another action potential. That's why this part of the action potential is called a relative refractory period, and this portion is absolute refractory period. And this part is relative refractory period. This can take second stimulus if the strength of the stimulus is strong enough. But whatever the strength is, if it falls within the absolute period, it will not produce any effect. That's why when one cardiac muscle contraction is going on, so by this time if you give second stimulus, it will not respond. এটা যদি নিত তাহলে what would happen? Cardiac muscle কে আমি খুব fatigue করে দিতে পারতাম. It was possible to make the cardiac muscle fatigued. Okay, we make question. It is cardiac muscles are not titanized. Cardiac muscles are not fatigued. Why? Because of long refractory period. Okay, so this sort of action potential is happening in what type of cardiac muscle? Working cardiac muscle. Let's see the difference. What happens in SA node? Cardiac, we have seen contraction of the working cardiac muscle needed a stimulus. But our SA node, AV node, Purkinje's fibers, they don't need any stimulus. They can produce their own impulse. Nobody has no requirement of any stimulus. So they can produce their own response. They can produce their own impulse, cardiac impulse. How? Now we'll discuss it. How many phases are there? There? 5. He said 5. You are telling 4. I am confused. Huh? Phase 0 is not any phase. Phase 0 is also a phase because there is an entry of sodium ion. So you have to consider it. So there are how many phases? Again 4.
No, five.
5. Okay, there are 5 phases. So now let us see what is happening in SA node. This is the mechanism of generation. Once it is produced, once this is produced in P cells, what is produced? This is nothing but an action potential. This potential is termed as cardiac impulse. Now this cardiac impulse from SA node will be traveling to the junctional tissues, through the junctional tissues. Okay. Now see, generation I got, how impulse is generated, now how it will be transmitted. Okay. So SA node is located where? I am having still 15 minutes. Where it is located? In right atrium. Okay. Just below the entry of superior vena cava in the posterior wall. So here it is being generated. Okay. Now suppose this is the SA node. This area is the SA node. So this impulse is generated here. Now what will it will transmitted? There are auricular muscles, cardiac muscles. Now it will transfer this impulse to the auricular muscles. This auricular muscle will transfer to the next auricular muscle, to the next, to the next, to the next, to the next. So they will be spreading like this. Ultimately, this impulse will be coming to the right atrium, from left atrium, from right atrium to the left atrium, and they will also, this impulse will also proceed to the AV node. While this impulse is traveling to the through the muscles of the atria, when they are coming towards the AV node, what happens? It is already having this impulse. How? It got this impulse by another route before it spread it to it, AV node. How it came to the AV node? There are special internodal tracts. There are 3 internodal tracts. So their purpose is to bring the impulses to the AV node. Okay, we know what is the conductivity of this internodal tracts? 1 meter per second. In 1 second it will travel 1 meter. Whereas these tissues, means auricular cardiac muscles, their conductivity is 0.3 meter per second. So they transmit slowly. Whereas this tract transmit the signals, accelerometer signals to the AV node relatively faster. That's why you get the impulse faster. Okay, this way AV node got. Okay, another important thing is that atrium and ventricle, they are 2 separate syncytia. They are 2 separate syncytia. What is syncytia? Who can tell? Yes, what is syncytia? Syncytium. Syncytia is plural, syncytium is singular.
The muscular tissues, they work as a single unit. The whole of atrium and the whole of ventricle works as a single functioning muscular unit.
Yes. See, tomorrow I will explain the morphology of cardiac muscles. The cardiac muscles, suppose she is one cardiac muscle, she is both are connected to each other. Not only this, she is also connected to her. She is also connected to her. All are interconnected. Okay, and having connection, cytoplasmic connection through what are there in the intercalated discs? Gap junctions. So since gap junction means the cytoplasmic connection is there. So because of this, if one cell is stimulated, all the cells become stimulated. So all the cells work as a single unit, that is syncytium. So auricular muscles work as a single syncytium, contracts at a time. But this impulse, there is no continuity between atrial muscle and ventricular muscle because both the muscles are separated by a fibrous layer. What is this? What is there in between? What is the name of this? Sinus, coronary sinus. So there is a group coronary sinus. It is made up of fibrous group. So because then what is the continuity? There must be a continuity between the atrium and ventricle. Who makes this continuity? Electrical continuity. It is the AV node, a bundle of His. It is only through the bundle of His electrical continuity is there between atrium and ventricle. Remember this. Okay? If there would have been continuity, if this there was no fibrous cord, what would be the result? Atrium, ventricle will have with simultaneous contraction would have been there, but normally it doesn't happen. First atria contracts. When its contraction is finished, then only ventricle contracts. Otherwise, contraction of the atrium leads to sending of blood to the ventricle. তাহলে দুটোই যদি কন্ট্রাক্ট করে সাইমালটেনিয়াসলি তাহলে এই ব্লাডটা যাবে কোথায়? কোথায় যাবে? সো যাওয়ার জন্য ওকে contract, coming the blood here. So now you contract. It is already now filled up with blood. Now you contract. So I must develop a system so that both are not contracted simultaneously. So what to be made? The impulse that is coming from SA node, I have to make some delay to allow completion of atrial contraction. When it is contracted, then by this time I will be sending it to the ventricular muscle, completion of atrial contraction, then so that ventricular muscle can contract. So there is a junctional delay in AV node. What happens in, not AV node, in bundle of His. In bundle of His, what happens? This bundle of His from AV node is located in right atrium. From here it is trying to enter in ventricle. First, it has to cross the fibrous cord. So while passing through this fibrous cord, what happens? These junctional tissues become very thin and coated by the fibrous collagenous cells. So they become very thin. We know thin wire and thick wire, who will be passing current more easily? Thick or thin? Thick. The conductivity will be more in thick. Okay. So since it has become thin, so its rate of conduction will be slowed down. So because of this change, there is delay of conduction of nerve impulse to the bundle of His. So now bundle of His will enter into the septum. The septum, suppose this is the septum, intraventricular septum, started here. It is the junction, atrium, ventricle. So it is the junction. So first, initial part of the septum is made up of membranous tissue. So first this will pass through this membranous tissue, then only it will enter in the myocardium. Septum is made up of myocardium. So septum is thick. So first most part of this bundle of fluids will be at the left side. Of the septum. Then it will be divided into two branches. One is left bundle, one will be coming to the right as right bundle. Right bundle is thin, left bundle is thin. As a result, left bundle, the septum, suppose this is the septum, here what happens? The outside means left side gets the impulse first and the right side gets impulse later. So there will be flow of current from that side to this side of the septum. So this will obliquely, which part question comes, which part of ventricle gets cardiac impulse first? Septum. Which part of the septum, left or right? Left. I said the bundle branch, which is thicker? Left. So this part will be getting first. Then to the right. So that's why current flows from left to right of the septum. In this way, ultimately it will reach to the apex. So ultimately, it is going to the apex through this septum. The impulse is going to the apex. Now from the apex, it will be spreaded through all the walls. This wall, that wall, this wall, the bottom posterior wall, it will be spreading towards the base of the ventricle. So this is the base of the ventricle. So from the apex, it will be spreading towards the base of the ventricle. And it is the last part, base of the heart is the last part that gets cardiac impulse. So this is how electrical impulse is generated, cardiac impulse is generated as a node and transmitted to the cardiac muscle. Is that clear to you now? Any question? You can expect question like this. How cardiac impulse is generated in a node? What is the mechanism? So it is produced by slow process, slow action potential. When it is produced there, now it will be conducted through the junctional tissues. So what are the routes? These are the routes. Ultimately, through Purkinje fibers, they will be distributed to all the ventricular muscles. First, we will get atrium, then we will get septum, then we will get apex, then we will get gradually to the base. So this is the conduct. Can you remember this? How it is generated? So this is all about this. Next day, we will be, in this class, we will be discussing about cardiac cycle. Very important.
So today we are going to study the effect of repeated stimulus in a simple muscle twitch. The effect of repeated stimulus. So for this, what are the materials required? It is written here. We need a nerve muscle preparation. We need a chymograph. Similarly, what we have done in our earlier classes, the requirements would be similar. Just here, the different thing would be, in earlier classes, we used to give one single stimulus. But now we are giving multiple stimulus and we are trying to see the effect of multiple stimulus on this muscle, on this curve created by the muscle twitch. We have to make sure that the electric shock or the stimulus which we are giving on the muscle on the sciatic nerve to excite the muscle should be of the same caliber. It should be the voltage should be same. There should not be any difference. So what exactly happens when you do this? See, firstly, this one is stimulus number 1. This one is stimulus number 2. This one is 3. You can see this now. Everyone can observe this. What is written on the graph? What is the name of the stimulus here? So you can see there is some difference in the latent period also, and there is some difference in the height of the curve also, and then there is some contraction remainder here. There is some difference in the relaxation period also. So how is this coming out? So first we can see here we are giving the first stimulus. The first stimulus is this. Then you can see in the second. Then you can see in the second and third stimulus there is the increase in the curvature. There is increase in the height of the curve. Why does this happen? Because the force of the contraction is increased. So what are the reasons for this? So first stimulus we are giving, the height is going up to a certain extent. Then up on second and third stimulus they are going a little high. So what is the reason behind this? So first one is there is increased release of calcium from the sarcoplasmic reticulum. So the calcium released from the sarcoplasmic reticulum, they will bind to the actin myosin and they will cause the contraction. So up to a certain period there will be extended release of the calcium ions. So there will be increase in the contraction of the muscle. Then there will be, at first the muscle is at rest, so we are giving one stimulus. Then on repeated stimulus, we will take up more ATP. There will be production of more and more ATP. So due to production of more ATP and due to availability of the nutrients, the carb will go up. The carb will go up means the muscle contraction, the vigorous contraction, the vigorosity of the contraction will increase. Then one thing we have already studied about temperature. So once we are exciting the muscle, once the muscle is twitching again and again, the temperature will increase. So this in turn will reduce the viscosity of the muscle. So once the viscosity is reduced, the liquid is free, the muscle is free now. So obviously the force of the contraction will increase. That is up to a certain extent. Then last thing which causes this increase, this increase is the abundance of the acetylcholine or the neurotransmitters. We know that when we excite a muscle, when we give a stimulus, there will be increase in the neurotransmitters. They will keep on, the body will keep on releasing, the muscle will keep on releasing the neurotransmitters. So there will be an increase in the muscle contraction. Then after 1 to 3, now this is 4. So after 3, you can see that there is a sudden drop in the carb. Means the contraction, the strength of the contraction has reduced. Here it has increased up to a certain time, up to 3. Then the fourth contraction has reduced. So in this case, why it is reducing? The things which have occurred in the first place, now the reverse will happen exactly. Like first what I have said, there will be increased release of calcium ions from the sarcoplasmic reticulum. The calcium ions are stored in the sarcoplasmic reticulum. They will get released from that part and they will go and bind to the actin myosin. So now the reserves are getting depleted. The calcium ions stored in the sarcoplasmic reticulum, they are getting less. So the calcium which is getting collected in the outside the sarcoplasmic reticulum, they are in this place the calcium concentration is increasing. And this release of calcium from the sarcoplasmic reticulum for the contraction to occur, this is a passive process because the outside concentration of calcium is less and the inside concentration is high. So this is a passive process. So this occurs rapidly. But again, what happens when the calcium ion, when it's time for the calcium ion to go back to the sarcoplasmic reticulum again. So after coming, it will go back again. It will replenish the stores. So again, when the calcium ion will go back to the sarcoplasmic reticulum, then this is a active process which requires ATP. But as we are using the muscle, as we are giving the stimulus, various stimulus to the muscle rapidly, so the ATP is going to be used up. The ATP, the nutrients, they are going to be used up. So due to the depletion of ATP, as the muscle contraction is also an active process, this requires ATP. And for the calciums to go into the sarcoplasmic reticulum, this is also an active process. So these things will be hindered. Due to this, there will be decrease in the power of the contraction. And again, there will be lactic acid build-up. And we have already seen in that room now what we have done with our muscle contraction, with human muscle contraction. There will be rapid build-up of the lactic acid and other waste products which will be getting accumulated because they cannot be removed in such a fast rate. As we are giving stimulus one after another in very rapid rate, so they cannot be removed so fast. So this will hinder the power of the muscle contraction. The muscle contraction, the power will be less. Then what are other things? So this is the reason why the graph will gradually decline. Now there is another thing called contraction reminder. Like see, first one when we are doing this one is 1, it is going gradually up to this. The contraction, firstly when we are doing the contraction, then the relaxation will be at the same rate. But at later stages what will happen, once we are doing contraction again and again, the muscle cannot fully relax. Because for fully relaxation of the muscles, again the calcium ions has to go to the sarcoplasmic reticulum. So these things is not happening. That's why there is a contraction reminder. So you can see the muscle is getting fully relaxed in this case. But here, after at 6 and 7 on repeated contractions, the muscle cannot relax properly. So here is the baseline. So you can see that the muscle relaxation has gone way above the baseline. So the muscle cannot relax properly. So again there is lactic acid build-up and there is use of ATP. So similarly for as ATP is required for the contraction, similarly for relaxation also ATP is also required. So due to depletion of ATP and the calcium ions not going back to the sarcoplasmic reticulum at a high rate, so that's why there is a contraction remainder, means the muscle cannot relax properly. And there is also a difference in the latent period. Earlier you can see that in 1 you can see here, then in 1 to 3 when there is an increase in the muscle power, there is also a decrease in the latent period. You can see now these things are closer to the point of stimulus. Like this one is suppose this one is the point of stimulus. We are giving the stimulus here. So there is the latent period. You can see the latent period is increasing with time. So these are the same things for which the latent period is also decreasing. So anybody has any confusion about this? The latent period will also be increasing on successive contraction, because the muscle, there will be more depletion of ATP, there will be build-up of the lactic acid and other waste products. So the muscle will have difficulty in contraction. So this will increase the latent period. That is the thing.